The overall goal of this standardized procedure is to screen water samples for endocrine active chemicals using commercially available receptor-based cell assays.
This method can help answer key questions in the water quality monitoring field, such as, are endocrine active chemicals actually present in the sample?
The main advantage of this technique is that it can analyze for a group of endocrine active compounds much faster than the current chemical methods.
This technique can help diagnose water quality issues that arise from the presence of endocrine disrupting chemicals.
This method provides insight into contaminants in water, but it can also be applied to other environmental matrices, such as sediment and tissues.
Generally, individuals new to this technique may struggle because it requires high precision when using pipetting techniques.
To begin this procedure, prepare the water sample and stock solutions of assay-specific medium and reference chemical as detailed in the text protocol. Store the assay-specific medium at 4 degrees Celsius for long-term storage. Next, add 15 microliters of the appropriate reference chemical stock to 285 microliters of assay medium in a sterile tube. Mix the sample by pipetting up and down. Then, add 200 microliters of 5% DMSO in assay medium to 8 additional tubes. Transfer a 100 microliter aliquot from tube 1 to tube 2 to begin a 3-fold dilution series. Mix the diluted sample in tube 2 thoroughly with a pipette. Then, transfer a 100 microliter aliquot from tube 2 to tube 3. Continue this process for each tube.